Good evening, and welcome to monobiology. This evening we're going to look at meiosis, which is the creation of sex cells. The purpose of meiosis is to form gametes. Gametes is the scientific term for sex cells. As you can see in the picture, we have parent cells, and we end up with 4 daughter cells. Notice the number of chromosomes in the parent cell, which equals 4, is reduced by the time we get to the end, and each cell only contains 2. This is important because, for example, with humans contain 46 chromosomes. Human cells contain 46 chromosomes, and if a sperm cell that had 46 chromosomes met up with an egg cell that had 46 chromosomes, then the resulting zygote, or fertilized egg, would contain 92 chromosomes, which is far too many for a human. So meiosis is necessary to create haploid cells, which contain half the number of chromosomes, from the original diploid cells, which contain the correct number of chromosomes. 46 for humans. In this case, this parent cell will be 4. We're going to take a little journey through the different parts of meiosis. Meiosis involves 2 phases, meiosis 1 and meiosis 2. And they're very similar, you'll find, to mitosis, although there are key differences that we'll highlight along the way. Meiosis 1. The key point that you need to be aware of for meiosis 1 is that diploid cells, which contain the full amount of chromosomes, are going to be turning into haploid cells, which contain the required number of chromosomes for sex cells, which just so happens to be half the original chromosomes. Notice that the phases of meiosis are very similar. Where we had prophase, metaphase, anaphase, and telophase in mitosis, we have prophase 1, metaphase 1, anaphase 1, and telophase 1 in meiosis 1. And that's a key difference. If you're asked a question about meiosis, you must refer to the phases with respect to the 1 for meiosis 1, or as we'll see later, the 2 for meiosis 2. Because prophase, metaphase, anaphase, and telophase refer specifically to mitosis. So make sure, if you're ever answering questions about meiosis, that you include the 1 or the 2 for meiosis 1 or 2, if that's what you're supposed to do for those questions. Now let's just take a look here at the different parts. Prophase 1, you'll find that the same kinds of things are happening in prophase 1 as were in mitosis. So you can see here that the nucleus is starting to dissolve, the centriole pair are moving towards opposite poles, and the spindle fibers, which you can just barely notice here in these pictures, are starting to develop. One thing that you'll notice, which is a little bit different in prophase 1 of meiosis than mitosis, is that you have chromosomes, but they're condensing like they did in mitosis, but in this case they're condensing next to their homologous partner. Okay, so you can see here we have a set of chromosomes. Notice how they're overlapping right there, and right there, and right there. All right, those overlaps are called crossing over. It's where a piece of each chromosome switches with its homologous partner. Okay, and you can see that evident in metaphase. See how these genes on these chromosomes have swapped. You see the darker one here on the lighter chromosome and the lighter piece here on the darker chromosome. That's as a result of what happened here, called crossing over. Okay, so crossing over, which creates genetic variety, is something different about meiosis that doesn't occur in mitosis. And we'll talk a little bit more about crossing over later on. Let's look at metaphase 1. You'll notice again that the chromosomes, it's very similar. You've got the chromosomes lined up in the middle, but one key difference that you probably have already noticed is that the chromosomes line up not single file in the middle of the cell, but next to their homologous pair. So you have them lined up in 2s in the center. Notice that the spindle fiber has attached to the centromeres of each of the chromosomes, which is important, and the centrioles are at opposite poles, and the spindle fibers are fully formed. In anaphase 1, we have the separation not of the chromatids, like in mitosis, but of the homologous chromosome. See? One homologous chromosome is going towards one, and its homologous partner is going towards the other end of the cell, and same with these two here. At the end, in telophase 1, the same things happen, except for at the end of telophase 1, we now have 2 haploid cells, as opposed to 2 identical cells. Neither one of these cells is genetically identical. And certainly neither one of these cells is genetically identical to the parent cell that we started with back in prophase. So again, to reiterate, key point here. Turning diploid cells into haploid cells. That's what meiosis 1 does. Meiosis 2 is simply duplicating the haploid cells. So if you look at these, again, we have prophase 2, metaphase 2, anaphase 2, and telophase 2. But essentially, if you look at these in a little bit closer detail, what you'll notice is that each of these haploid cells is simply going through mitosis. Okay? You see between prophase 2 and metaphase 2, see they're lined up single file again. The centrioles are there. The spindle fibers are attached to the centromeres. And here the chromatids are being pulled apart. And we end up with the only difference here between meiosis 2 and mitosis is the fact that we end up with 4 genetically different. Take a look at each of these cells. We end up with 4 genetically different cells. And that's key. And of course we started with 2 genetically different cells, so it's not surprising. But essentially all meiosis 2 is mitosis of the haploid cells. Now, what you end up with here at the end of telophase 2 depends on 2 important things. First of all, crossing over, how many times it occurred, whether or not it occurred. And there's another factor that occurs during metaphase 1 of meiosis, which is independent assortment. Okay, and that is essentially the random lining up of homologous partners with respect to each other. And we're going to talk more about that in greater detail later on. But in terms of meiosis 2, again, it's PMAT, prophase, metaphase, anaphase, telophase 2. And essentially all it is the duplication of the haploid cells, or the replication of the haploid cells. Notice it says here, and this is important, that there's no interphase between meiosis 1 and meiosis 2, and there is no DNA replication and no growth of the cell between those two meioses. So meiosis 1 and meiosis 2 pretty much happen right after one another, and there's no interphase in between them. All right, to sum up here, meiosis is the formation of gametes. This involves the formation of a haploid cell from a diploid cell, haploid cells from a diploid cells. In the process of turning diploid cells into haploid cells, 2 processes known as crossing over and independent assortment add genetic variety to the cells. This ensures that no 2 gametes or sex cells are identical. Just to reiterate, remember, if this process didn't occur, then unfortunately when this sperm met this egg, you'd get the incorrect number of chromosomes going in. So it's very important that a sperm cell that has 23 chromosomes matches up with an egg cell that also has 23 chromosomes. So we end up with 46. And that in a nutshell is all you need to know about meiosis. Well, not all you need to know. We're going to talk more about crossing over and independent assortment a little bit later on. Thank you for listening.